Hello students, this is the second module on the various resistance mechanism in plants. So like I had told you in the previous video, we know that plants have different ways to defend themselves whenever a pathogen attacks them. And we have already spoken about the different structural defense mechanisms that are present or that are pre-existing in a plant. Today we will be talking about the different structural defense mechanisms that are shown or elucidated by the plant post infection or once the organism has or pathogen has infected the plant. So all the post infection structural defense mechanisms or defense structures that are developed is what we will be discussing about today. Now the structures that are developed after attack can be grouped under four different categories. We have histological structures, cellular structures, cytoplasmic defense structures and necrotic or hypersensitive defense structures. So we will be dealing with each one in detail. Under histological, we again have several types like cork layer, abscission layer, thylosis and deposition of gum. Let us start studying each one in detail. So the histological defense structures are formed due to differentiation of the tissue or due to deposition of some chemicals in the tissues in or around the pathogen attack site. When we have any changes in the tissue, that is, or any differentiation in the tissue, that is what we call as histological defense structure. So please remember, once the pathogen has attacked, then we have formation of each of these structures that we are dealing. So the first histological defense structure that is developed, or that one of the important ones that can be seen as a defense mechanism, is cork layer. Now cork layer is developed usually below the point of infection wherever the pathogen has attacked, and it is developed below the point of infection to prevent further spread of the pathogen. Not only does it prevent the further spread of pathogen, it also blocks the inward flow of any toxic substance that is produced by the pathogen. So even if the pathogen has entered inside, even if it produces some toxic substance, that substance will not go further into the tissue because of this thick cork layer that has been formed. Also, from the plant, there will be no flow of nutrients or water to the infected area, due to which the pathogen gets deprived and it gets killed easily. These are the reasons behind the formation of a cork layer by the organism or by the pathogen. So we can see over here, this is the cork layer which is marked as Cl. H is the healthy tissue and I is the infected tissue. Between the infected tissue and the healthy tissue, we have formation of a thick cork layer which is preventing any flow of nutrients from the healthy host to the infected tissue or any flow of toxic substances or spread of the pathogen this way. So this is the importance of cork layer in plants as a defense mechanism. Now this has been seen or this has been demonstrated in the soft rot of sweet potato, which is caused by Rhizopus species, or potato tuber disease, which is caused by Rhizoctonia, or even in case of tobacco mosaic virus, which causes tobacco mosaic virus in tobacco mosaic disease in tobacco plants. So in all of these examples, it has been seen that once there is infection by the pathogen, the plant tends to produce this cork layer as a defense mechanism. So this was the first histological defense mechanism, that is cork layer. The next histological defense mechanism is abscission layer. Now abscission layer is also a method that is used to separate the healthy tissue from the infected tissue, but here we are talking about empty space between the two tissues. So usually abscission layer is developed in a healthy plant to separate the ripe fruits or the old leaves from rest of the plant. That is the reason a plant usually forms abscission layer. But in case of attack by a pathogen, it forms this empty space or it forms this abscission layer between the diseased part of the plant and the healthy tissue of the plant by dissolving the middle lamella. So once the middle lamella has been dissolved, that empty area which is left out is called as the abscission layer. So what happens is the infected area or infected locus becomes unsupported. You can see over here, the abscission layer is the empty space which is separating the diseased area from the healthy area. Now this leads to the diseased area or infected area becoming unsupported. It will finally shrivel and ultimately fall down with the pathogen in it. So that is helping the plant to check the spread of the infection. This is seen mainly in case of stone fruit trees. In fact, this is a picture of a leaf that is seen in the plum plant. So here you can see the abscission layer has been formed to check the spread of the pathogen. This is mainly seen in case of this is even a disease that is called as short hole disease, which is caused by Stigmina carpophylla, and also in other stone fruit trees which are affected by the pathogen Xanthomonas pruni, we have formation of abscission layer. So cork layer was one of the examples of histological defense structure. The second one is abscission layer. The third histological defense structure is tylosis. Now tyloses are outgrowths of protoplast of the parenchymatous cells which will protrude into the xylem. Normally this will obstruct the flow of water. So in case of a diseased plant, not only water flow, it will also obstruct the spread of the pathogen. This is seen commonly in case of fusarium wilt, which is caused by Fusarium oxysporum. Now here I have given you example of wilt of sweet potato, which is caused by Fusarium oxysporum, Forme specialis batatas. So in case of this organism, it has been seen that once the pathogen attacks the plant, there is formation of tylosis. You can see over here, these arrows which are pointing are the tyloses. So in the xylem vessels, we can see that there are outgrowths of the protoplasts of the neighboring parenchymatous cells, which is going to block that entire channel, or which is going to obstruct the flow of the pathogen and the flow of the water. These outgrowths of protoplast are called as tylosis. Tylosis is a very common defense mechanism seen in case of wilt diseases. The fourth histological defense structure is gum deposition. Now, gum deposition mainly serves as a method of demarcation or as a method of protective demarcation, and it is seen around the lesions which are caused by the pathogen in case of this organism, that is Magna griseae, which causes blast disease of rice. So in this organism, it is seen that wherever there are lesions, to prevent further spread of the pathogen, around the lesions gum is deposited or gel-like substance is deposited, or it can also be deposited in the parenchyma cells, in the xylem cells and the intercellular spaces, which is seen in case of the organism or pathogen that causes black rot of apple, that is Physalospora sidoniae. In case of this pathogen, it is not only around the lesions, it is also in the parenchyma cells, in the xylem cells and the intercellular spaces which is found between the cells. So deposition of thick polysaccharides or deposition of gum mainly helps the organism again to check the spread of pathogen. You can see over here, this is an aspen plant. Again here you can see this arrow shows you tylosis, whereas this arrow head shows you deposition of the gel or deposition of the gum. So these are the four histological defense structures which are developed in response to attack by a pathogen, that is cork layer, abscission layer, tylosis and gum. The next method of defense or the next structural defense mechanism which is induced in response to the pathogen attack is a cellular defense mechanism. Now cellular defense structures are actually not very effective as a defense structure. They are not very effective in blocking the pathogen, but they are also one of the methods of defense that are shown by the plant. So under cellular defense structures, we have two types of structures. One is swelling of the cell wall. Now it has been seen that the epidermal cells and the subepidermal cells, when they come in contact with the hyphae, their cell wall gets swollen up to inhibit further penetration of the pathogen. So only the epidermal and subepidermal cells cell wall has been shown to swell up to inhibit further penetration. This has been seen in case of Botrytis cinerea, in case of pea leaves, and Cladosporium cucumarinum, which is causing infection in cucumbers. So it has been seen that in both of these pathogens cases, the cell wall gets swollen up. The second cellular defense structure is sheathing of hyphae. Now this is again due to changes in the cell wall. So the cell wall starts stretching inwardly. This is mainly seen in case of fungal hyphae. So it has been seen that the fungal hyphae get enveloped due to the inward stretching of the cell wall. You can see over here the fungal hyphae are getting sheathed or they are getting covered, getting enveloped due to the stretching of the cell wall in an inward fashion. So Usually the hyphae, even despite this sheathing that happens, even despite this covering that happens, usually the hyphae manage to penetrate further and enter into the plant, as you can see over here. Still, this is considered as a defense mechanism because the sheath or the envelope formation delays the penetration by the pathogen. That's all. So that's why I told you in the beginning as well, cellular defense structures are not very important in defense, or they are not very effective, but they do delay the penetration, or they can delay the spread of the pathogen. So the two cellular defense structures are the cell wall swelling up and hyphae sheathing. The cell wall gets swollen up, and when it comes in contact with the hyphae, which can prevent the spread of the pathogen, and hyphae can get covered due to this swelling, or it can get covered due to this stretching of the cell wall. This is seen in case of these particular organisms. The third type of defense mechanism are cytoplasmic defense structures. So we have spoken about histological and cellular defense structures. Now we are going to discuss about the cytoplasmic defense structures. This too is effective. This type of defense structure is also effective only against slow growing or weak pathogenic fungi. What happens over here is, after the entry of hypha into the cell, so once the fungal hypha enters inside, penetrates inside the cell, the cytoplasm becomes granular and dense, which leads to disintegration of the hypha, the fungal hypha, into smaller granular bodies, eventually stopping their development. So the cytoplasm of the host cell itself becomes granular, due to which even the penetrating hypha gets disintegrated. That is the cytoplasmic defense mechanism. The last one is the hypersensitive response, also abbreviated as HR. Hypersensitive response is a type of a programmed cell death, and it is very common in case of biotrophic fungal parasites or in viruses and even in nematode infections. Now here, the infected host cells and the cells which are in the vicinity, so not only the cells which are infected, even the ones which are around it in the vicinity will die rapidly. That is why it is called as hypersensitivity, because they react in a hypersensitive fashion or in a hypersensitive manner. So the infected host cells and the cells which are present around the infected host cell will die very quickly, or they will show a hypersensitive response. Though the actual mechanism of this hypersensitivity is biochemical, it is being considered under the structural mechanism, because when the hypersensitivity occurs and when the cells start dying, that entire dead tissue or the necrotic tissue, necrotic is the dead tissue, that entire necrotic tissue is the reason the pathogen is not able to spread, or that is the reason which checks the spread of pathogen. That is why it is being considered under a structural defense mechanism. So even though hypersensitivity occurs due to biochemical reactions inside the cell, ultimately that entire area gets dead or necrotic, which is why the pathogen is unable to move further. That is why it is considered under a structural defense mechanism. So hypersensitive reaction or response is being noted in, or it has been seen in case of Syncytium endobioticum. Syncytium endobioticum causes wart disease of potato. Phytophthora infestans causes late blight of potato, and Magnaporthe griseae, which causes blast disease of rice. So in these organisms, it has been seen that when they infect their host plant, the host plant shows a hypersensitive response. Now what happens in this response? So in this HR or hypersensitive response, the fungus, now this is the fungal spore. The fungal spore will come in contact with the host protoplasm. So you can see over here, this is the host cell and this is the host cell nucleus. The fungal spore will now try to enter into the host cell. When it enters into or tries to enter into the host cell, it will come in contact with the host protoplasm. So it has penetrated the cell wall and it will come in contact with the protoplasm. This will trigger the host nucleus to move towards the pathogen. So you can see over here, the host nucleus is slowly moving towards the pathogen and it has come near the pathogen. The moment it comes near the pathogen and simultaneously the pathogen penetrates inside, you can see over here the infection peg has penetrated inside. The moment this happens, the nucleus starts disintegrating into granules. So you can see here the nucleus disintegration has started. Now when the nucleus starts disintegrating into granules, these granules will accumulate around the pathogen. So you can see here, whatever degranulation or disintegration has happened here, those granules are now accumulating around the pathogen. And once they accumulate around the pathogen, later they do disperse in the cytoplasm, but once they accumulate around the pathogen, this leads to the cell membrane swelling up. So you can see over here, the cell membrane has also swollen up and the granules have dispersed in the cytoplasm. Eventually, the cell is going to die. Now, if the cell dies, the pathogen which has infected that cell also gets degenerated. This is the mechanism of hypersensitive response. So, all of these responses are triggered by pathways, or they are triggered by signals which are biochemical. But in this manner, we have several cells in a tissue getting dead or necrotic, which is why it is considered as a necrotic response or a hypersensitive response, and it is considered under the structural defense mechanism. So hypersensitive response is a very important defense mechanism. It is, like you can see, an example of a programmed cell death. So the moment the moment the host realizes that the pathogen is coming in, it kills itself, and in this way, even the pathogen is getting nullified, or even the pathogen is getting degenerated. So these were the structural defense mechanisms which are formed in response to attack by a pathogen. Under the structural defense mechanisms, till now what all have we done? In the previous video, we had spoken about or discussed about the pre-existing defense structures, which included the cuticular wax, structure of the epidermal cell wall, structure of the natural openings, and some internal structural barriers. These are pre-existing in every plant, or in the plants I gave you examples about. Structures which are developed in response to the attack by a pathogen are what we have dealt today, which includes the histological barriers, cellular barriers, cytoplasmic barriers, and hypersensitive response. Under the histological barriers or histological structures, defense structures, we have discussed about cork layer, abscission layer, tylosis, and deposition of gum. Under the cellular defense mechanism, which I told you is not very effective, we had swelling of cell wall and sheathing of hypha. Under the cytoplasmic defense structures, which again is not a very effective defense mechanism, we had the cytoplasm getting granular and dense and disintegrating into, which led to the disintegration of the hypha, the pathogen hypha. And the last one is the hypersensitive response, wherein the tissue gets dead, or the tissue is necrotized, the tissue is killed because of a hypersensitive response by the host cell. This leads to the pathogen also getting degenerated and acts as a resistance mechanism or a defense mechanism. So we have finished with the structural defense mechanisms. In the next class, we'll be talking about the biochemical defense mechanism which a plant has that will help it to evade the pathogen. I hope you all have followed today's class. Thank you.